Now let's look at the regulation of iron homeostasis in the body. This is basically the regulation of iron levels. Iron levels need to be maintained very closely because disposal of iron from the body is not readily available. So typically a bleed is the only way to get rid of iron. So the amount of iron absorbed into the body needs to be controlled. And this is largely controlled by a protein that is made and secreted by the hepatocyte. So let's take a look at this pathway. The hepatocyte, of course, is storing iron as it is bound to ferritin inside the cell. The level of iron inside the cell, along with communication from bone marrow using bone morphogenic protein involved in erythropoiesis, is going to be the sensors and regulators of iron levels, both in the body and how much iron is released by the liver. So recall that in order to deliver iron to the blood to bind to transferrin, we have a transporter called ferroportin. The level of iron release and iron uptake in the body is controlled by a small polypeptide hormone called hepcidin. Hepcidin is made by hepatocytes, and its protein synthesis is controlled by iron levels and communication from the bone marrow. So as more iron is needed, hepcidin expression will decrease. As more iron is stored, hepcidin expression will increase. So how does hepcidin affect iron levels in the blood? Hepcidin, when it binds to ferroportin, induces internalization plus degradation of ferroportin. So hepcidin binding to the ferroportin transporter induces its internalization and degradation. Once that happens, the iron that is located bound to ferritin inside the hepatocyte cannot exit the hepatocyte and enter the blood. So thus, this will control the release of iron from the liver, from the stores of the liver. But what about dietary iron? If we have sufficient iron levels in our body, such that we are now synthesizing hepcidin to block ferroportin release, then maybe we want to control the dietary uptake of iron. The other video on iron uptake and homeostasis shows the mechanism of dietary iron into the intestinal enterocyte. This mechanism holds true even in cases where we have high hepcidin levels and plenty of iron stored in the body. But recall that in order to bring iron from the inside of the enterocyte after it has been taken up to bind to transferrin in the blood and be carried to the liver, we have to use the transporter ferroportin. Hepcidin has the same effects on the ferroportin transporter on the base of the intestinal cell as it does on the surface of the hepatocyte. Hepcidin will inhibit iron release. Therefore, the iron will be stored as it's bound to its storage protein, ferritin. Now we know that iron overload is toxic. So what happens to the intestinal epithelial cell is that, remember, approximately 1/4 of the intestinal epithelial cells are shed per day. So the intestinal epithelial cell will continue to take up iron by its normal mechanism, but it's the transport of the iron into the blood which is inhibited and controlled by hepcidin. So therefore the iron accumulates inside the cell, but the cell is shed, it's sloughed off eventually, and it's discarded in the stool, and then the iron is not taken up into the bloodstream. Now there's two important factors to recognize and identify with the ferroportin hepcidin control system. The first one is hemochromatosis. This is more commonly known as iron overload. Individuals that have hemochromatosis have large iron deposits in their livers. Iron, of course, in high concentrations is toxic, and so hemochromatosis, if uncontrolled, can cause extensive liver damage. So what is going on with these individuals? Well, it's thought that these individuals that have this disorder have a genetic error in their gene for hepcidin. This genetic error leads to significantly reduced expression of the gene. So we have significant decreases in circulating hepcidin. Now what this effectively does is it impacts our regulation mechanism of uptake of dietary iron, so that whatever dietary iron is taken in, the ferroportin transporter remains on the surface, and then the iron can pass. So without hepcidin control, we have continuous dietary iron intake. The transferrin transporter system is still functional, and so the hepatocyte that is going to store the iron is still able to continually accumulate iron because of this functioning transporter system with transferrin. This system is not regulated, except in the fact of transferrin expression. So what we end up having here is large amounts in hemochromatosis of our ferritin iron complex, so the iron storage is dramatically increased. The other aspect of this is what is termed anemia of chronic disease. And the chronic disease we're talking about is any diseases that lead to chronic inflammation. Chronic inflammation, producing effectors such as TNF-alpha, interferon gamma, etc. These cytokines and inflammatory mediators are able to regulate hepcidin expression. And they regulate this in a matter of dramatically increasing hepcidin expression. So what does this do? If we have significant amounts of hepcidin, we are going to downregulate ferroportin. So we are going to dramatically decrease ferroportin. On the cell surface. Now what that's going to do is, of course, it's going to prevent dietary iron from transporting from the interior of the intestinal epithelial cell into the blood with transferrin. So this will block dietary iron uptake. And look what it'll also do to the iron stores in the liver. It will block release of iron from liver storage. So although we will have significant amounts of iron stored and bound with the ferritin complex in the liver, the iron is not able to transport because ferroportin has been degraded due to the high levels of hepcidin. So even though there's sufficient iron, this leads to anemia because the iron is unavailable as it is bound to ferritin inside the hepatocyte.